Good afternoon, OTS community, and welcome back to the Next Gen Early Career Scientist session. My name is Crystal Johnson, and I just graduated from David Corey's lab at UT Southwestern.
And my name is Jillian Belgrad. I'm an MD PhD student in Anastasia Kurova's lab at UMass Chan Medical School.
Jillian and I are so excited to welcome you here together, to be with you and welcome you to this session that's really important to us. This session provides a unique platform for student voices to be heard, and we have a great set of talks for you all to enjoy from students from all over the world.
At the end of each talk, there will be 5 minutes for questions, and given that this is a session by trainees, for trainees, featuring trainees, we invite everyone, but especially trainees, to come to the microphones at the end of each aisle, and feel free to come to the microphones even as the speakers are wrapping up to save time and we can get through everyone's questions. And without further ado, we are thrilled to introduce our keynote speaker for the session, Dr. Taavi Leto from University of Tartu in Estonia. Dr. Taavi Leto is an assistant professor, and before beginning his lab, he studied pharmacy and then got his PhD in biomedical engineering, both at the University of Tartu in Estonia. Following his PhD, he went on to do multiple postdocs across Europe in France, Germany, and Sweden, and then went back to his alma mater to begin his research group. One thing that we really wanted to make special, both in his introduction in this session today, is the fact that our career development and trajectory as trainees is really influenced by both the science and the people around us. So we wanted to highlight a couple key features that we asked Dr. Leto to share more about. That being that when asked about the most meaningful mentorship relationships, he shared that they were his academic advisors and professors along the way in each step of his journey. And then when asked what was the most meaningful and what's the most proudest accomplishment so far in his career, he shared that it's actually the fact that he's from a dual academia relationship with both he and his wife running prolific research programs at their home alma mater at University of Tartu. And so with that, we are thrilled to welcome Dr. Leto to the stage and begin his talk on peptide conjugated nucleic acids. Thank you.
Hello, everyone. Thank you very much for the very nice introduction. It's an honor to be in here at the OTS meeting and to be able to present at this junior researcher part of the program. Okay, like it was said, I'm Dr. Taavi Leto. I will talk about the peptide-mediated delivery of RNA therapeutics. Also some interest slides. So some introductory to what are the delivery peptides as such. I mean, the cell-penetrating peptides are cationic and or amphipathic peptides, usually up to 30 amino acids in length. They enter cells via endocytosis, so endosomal entrapment is a key feature also for these types of molecules, similarly to other non-viral vectors. They can generally, I mean, the main strategies how you could vectorize with CPPs are that you can do covalent conjugates with the cargo molecules, or the other option is the so-called nanoparticle formation approach. So which types of molecules are usually delivered with peptides? I think it arises from peptides, proteins, to different types of nucleic acid-based molecules, and also a variety of RNA-based compounds. So my entry into the peptide field started when I joined Professor Ülo Langel's group at the University of Tartu as a PhD student. He was sort of one of the pioneers in this CPP field. And while they have had, and he had had with his group quite some success in terms of using peptide conjugates with PNA, for example, then at the time, the sort of key thing that we wanted to develop was how we could convert this peptide-based strategy to be compatible with a nanoparticle formation approach. So what we did, we sort of thought that hydrophobic modification strategies would be worthwhile to use, and so we sort of started out with doing various fatty acid modifications on the different CPPs, and especially the stearic acid modifications that we used on the peptides was the sort of key modifications that turned out to be a good strategy line for achieving effective peptide-based molecules. So our screenings initially were done in this classical cold splice switching or splice correction assay. And what we could see, I mean, in this instance, I mean, in this nanoparticle formation approach, I mean, the peptides themselves without any modification usually are not active at all. And then with the stearic acid modifications, we were able to identify that the sort of one of the classical transportan 10, the transportan deletion analog that was previously developed in Nullos group, turned out to be a sterile TP10, turned out to be a good vector for short oligonucleotides. And sort of this was the first of the PEP peptides that we developed. And in a number of papers, we were able to show that these were effective for the delivery of short oligonucleotides, but also larger pDNA molecules, for example. Now, at the time, of course, everybody was also trying to find good vectors for siRNA molecules, like they do in some ways today as well, of course. And so what we recognized that, of course, we were also finding good vectors, and we were screening all of these modified CPPs to see if they would be active for the delivery of siRNAs. Unfortunately, none of them really were. What we recognized was that some of the molecules, I mean, they did form nanoparticles and were taken up by the cells. And if we were doing, for example, co-treatments with chloroquine, we could see that we were able to achieve RNAi activity. Now, by using this rationale, we were thinking that if we could sort of covalently attach something like chloroquine to our peptide vectors, and that was what we did. So we took succinylated trifluoromethylquinoline-based compounds and attached these to this transportan 10 peptides. And this was further modified also with the stearic acid modifications that I showed previously. And this part was essential in terms of conferring for the activity in the serum context. This PEPFX6 peptide, so to say, I mean, was nicely able to form nanoparticles with siRNA, and was also induced robust RNAi activity or gene silencing in a number of different cell lines. I mean, exceeding the activity of the sort of then present RNAi max and lipofectamine analog lipid-based transfection systems. So obviously, we also, I mean, were encouraged by that and wanted to see if this would also hold in the in vivo context. So we did, we're using the HBRT1 as a target, and we're injecting the formulations into mice, and we're able to show that we had gene silencing activity in a number of tissues, obviously most prominently in the liver. And we were further able to corroborate these results in several other models. And at the time, in principle, the compounds were as active as the hydrodynamic injection technique that was present at the time. And I think we're basically also in the range of the earlier lipidoids and lipid nanoparticles in terms of activity. So all in all, we actually did publish a number of these PEPFX papers and were able to show the delivery, like I mentioned, for the short splice switching oligonucleotides, the siRNA molecules, and also much larger plasmid molecules. And I think this PEPFX series of peptides have been followed up by more than 50, definitely 50 to 100 different papers in the following years. At that stage, also today, a little bit, I was asked to also like a little bit make like a view on how I have navigated my academic career. So I will a little bit also have like snapshots of where I have been from time to time. I mean, at that stage, I defended my PhD at the University of Tartu, and obviously in that stage, I've already realized that I would like to pursue academic career, so a postdoc was on the agenda for me. I was looking on two different directions at the time. One was to go to Sweden, and the other approach idea of mine was to go to Germany. But I had previously been collaborating with Bernard Lebleu, who approached me and said that if I would like to come down to Montpellier to work with him on a more special projects that he had at the time. And I had a small kid and wife and said that we can move to southern France for a while, and I think they didn't oppose much, and so we had a good opportunity to move to Montpellier, that is not far away from here. And there we were working with and also with Prisca Bogaran, who was a senior researcher in Bernard's group at the time. And then they had a larger project that was a collaboration with Matthew Woods and Mike Kate, who had developed these PIP peptides, or PNA or PMO internalization peptides. And I mean, in this space, sort of the part of the project that was relevant in Montpellier was to shed some light on how do these compounds behave intracellularly in the context of this different muscular models and also sort of in vitro DMD models. And so we did quite intricate uptake and trafficking studies by using skeletal muscle models and also primarily cardiac muscle models in terms of identifying how these compounds behaved in there. And what we were able to see in there was that actually in the more muscular models, cellular models, these PIP peptides were PIP6A PMO at the time, were highly active, while at the same time in the cardiac muscle it was much more complicated to achieve reasonable activity level. So the sort of differences that had previously been seen also in the in vivo context, and maybe I add also that these peptides were one of the first to really show cardiac activity in this Duchenne model. So this sort of barrier of having activity also seemed to be present in there. And then we were further sort of going deeper into the mechanism why this could be, and were able to show by using different endocytosis inhibitors that the sort of endocytic pathways that were responsible for their uptake in the muscular or cardiac muscle models were different, and caveola-mediated endocytosis was the sort of more or was the prominent uptake pathway in the muscular cells, skeletal muscle cells, and clathrin-mediated endocytosis was responsible for their activity in the cardiac model. So I mean, I think it's still a good comprehensive study on this, and was shedding some light how the differences in different cell types could have an impact on the activity of such compounds. Okay, at that stage, I had been previously in contact with Professor Ernst Wagner, who was also my opponent for my PhD, and we had been in touch for a while to be able to do a PhD or do the postdoc with him, and so the opportunity arose. So I moved to Munich, and we had a couple of papers in there in biomaterials and bioconjugate chemistry that were dealing with a sequence-defined polymer platform that he had developed, and also working on sort of folate targeting and in the context of siRNA therapy in cancer. And we were able to show pretty decent activity in different cancer models. But the sort of other project that I was really interested in there was that if we could do, like I showed with these PIP peptides, so the idea was that could we do something similar with this polymer, our sequence-defined polymer platform that Ernst had. So we set out to do that, and with Ernst, and then he also had Dr. Ulrich Lachelt, who was working at the time in his laboratory, and ultimately even Ulrich was the key guy who was driving this project. So what we came up with was this sort of polymer compound that was composed of these STP units, so sort of diaminoethan motifs that were endosomotropic, and then also, of course, had a PMO conjugated in there. And another important part was the sort of that it also contained double fatty acid modalities. And what we saw in our studies in the splice correction assays was that especially the fatty acid component had a really strong impact on the activity of these compounds, and we saw that the sort of some of the unsaturated forms, especially the linolenic acid sort of derivatives, had the highest activity in these models. And then further on, we also reached out to Yoshi Aoki's lab in Japan, and with the help of Joel Nordin at the time, who was a postdoc in there, they were also screening these modalities in this H2K MDX52 myotube model, and were able to show that that these molecules actually were quite potent and could induce strong exon skipping activity at relatively low nanomolar concentrations. And and then a few years ago, we were able to publish that in the Advanced Functional Materials Journal. Now, in the next stage, I had this, and maybe this is something that is also part of academic career, is that then I had to concentrate on my family. So I went to a parental leave for a year. Like it was said, I come from a dual academic family, so my wife, who was a PhD student at the time, needed to finish up her PhD, so I stayed at home with the kids, and she successfully defended. Defended in 2015, and then we had like a strategy that, okay, now we will go to postdoc together. And so our sort of next episode was that I was, of course, at home, not only or caring for kids, but was also writing some grant applications pretty heavily. And then we planned to go to Sweden, like I initially had in plan. And then, and I did my next postdoc with Professor Edward Smith, who is also in here, and Professor Samir El-Andalusi. And obviously, my wife also managed to get a really good PhD or a postdoctoral position at the Karolinska. So we moved to Sweden for the time being. And then at this stage, I was also writing quite some grant applications in Sweden in terms of, over some of these years to be able to start my own laboratory. And some of, one of these were successful. I got a grant from the government of Estonia to set up my own laboratory in 2018. So it was founded in 2019. And then in the last part of the study, I'm actually concentrating on some of the projects or a project that we started already at KI, and then with what we have been later collaborating with my lab. And this is also about peptides. So we had quite some previous knowledge on how to design peptides. So we thought that we will go and have another go and develop another platform for the delivery. So the basis for that was that we knew that the amphipathic peptides were important, and also that the helicity played a key role. Additionally to the sort of and some of these hydrophobic modifications as well. So, but in this context, so the design what we had was that let's design like a near ideal helical peptide with an amphipathic character, and so we did. And then also at the time, some of these peptide stapling chemistries were around, and we were considering, wanted to use this peptide stapling to sort of constrain these molecules into the alpha helical conformation. To our big surprise, when we were doing the screenings with these non-stapled, sort of peptides carrying just the sort of modifications, or also the stapled versions, were that also the non-stapled versions were active. So actually these alkenyl alanines that we used in there, these were hydrophobic enough to actually turn these peptides to be really active entities or something. And then we made derivatives of this peptide carrying increasing number of these compounds. And so one of our project was that if we could use these HPEPs now that we called from hydrophobic and also helical character, that if we could use them for the delivery of modified mRNA. And indeed, our initial sort of formulation optimization processes, I mean, we saw that we could really find conditions where we could have really small nanoparticles with the sounds of around 100 or below that, with a positive surface charge. And okay, when we knew that we had the nanoparticles at our hands, the next stage was that we hypothesized that would they be taken up by the cells by using the fluorophore-labeled mRNA. We were then, in the FACT studies, were able to see that we really had nice dose-dependent uptake of these compounds in the A549 cells. And then to provide like a more qualitative perspective, we also did confocal microscopy and were able to identify that we had really nice vesicular sort of distribution, which is reminiscent of the endothelmic uptake of these types of compounds. So they were effectively taken up and seemingly entered the cells by endocytosis. So in the next stage, we were looking at different cell lines. Would they be also functionally able to deliver mRNA molecules? And indeed they were. We screened a number of different cell lines, some of which, like HeLAs and C2, C12s, and A449 and HEC that I have shown in here. In all of these, we were able to functionally deliver mRNA with decent activity. So obviously the sort of next step was that would they also be active in the in vivo conditions? And indeed, we were using the FLUC mRNA as a readout model, and were able to show that we really had nice, I mean, and administrating the formulations by IV injections, we were able to show from the dissected organs that we really had nice dose-dependent activity in different tissues, including in the lungs, liver, and spleen, and also in kidney to an extent, and maybe the most prominently in the lung context. And so we had pretty decent activity in different tissues. Now, in the next stage, I think also one of the pharmaceutical companies or biotech companies, Evax Therapeutics, got interested in our platform and licensed it out from us, and also sort of provided financial muscle to carry out a little bit more complex experiments with that. And the plan with them was that we sort of validate this H-peptide platform also in the non-human primates or something. Now I mean, the formulations as such were relatively easy for the context of mice experiments. I think the sort of building up for the primate studies, I mean, yeah, provided much higher barrier in terms of also setting up the formulations and scale up. So what we did was that we had to actually implement the microfluidics production scheme for the formulations, which we were able to do, and also establish all the sort of downstream processing aspects and sterilization and storage conditions for these formulations. And ultimately, we were also had to, or we, with the help of precision nanomedicine in Canada, we could finally do the sort of final scale up and the production of this formulation. So the sort of target that we had in mind was the human EPO mRNA, which has been widely also utilized for validating different LNP platforms, for example, and we sought to take this as a model. The formulations were 70 nanometers in size with a relatively low PDI value of 0.17 and had a very high encapsulation efficacy of over 90%. And so we then were able to carry out this study in ITR in Canada, where the cynomolgus monkeys, they were injected with a long bolus injection. And then we were tracking and after the injections, obviously, we were checking if we would have EPO expression in the monkeys. So in the serum, we were able to see nice dose-dependent induction in the production of human EPO with a peak concentration at 6 hours. And then then we had another arm of the study, which was the sort of repeating dosing study, where we could see that if we were injecting the same monkeys on a weekly basis, then we could achieve the similar type of inductions from one week to another. So they didn't wear off that it has been a problem with some of these platforms, with some of the other platforms. And so, we were also looking at some of the side effects profiles, obviously, with these molecules. And the most prominently what we saw was that we have some complement activation upon these treatments in the monkeys. So with that said, I'm in the stage where I'm starting to conclude. So I think, like I've managed to show, that I think the peptide-based drug delivery systems like PepFX or HPEP peptides comprise excellent potential for the delivery of a wide variety of RNA-based modalities, including short oligonucleotides, modified RNA, or also in some contexts CRISPR-Cas systems, something that I haven't really shown today, but what we have been working on. And I also think that the peptide systems as a whole can offer novel means for the delivery to extrahepatic tissues, and hopefully can a little bit challenge the sort of landscape of delivery in the future. So with that, I would like to also acknowledge my own laboratory at the University of Tartu and number of collaborators in Tartu at the Karolinska Institute, in the Munich lab, and in Vienna, and okay, the laser is not, and also in Japan and in France. And with that, I'm concluding my talk and would be open to some of the questions.
So thank you, Tavi. I guess my question was, so I think in one of the slides you showed that these particles are taken up by endocytosis. Do you guys have an idea about what sort of receptors or mechanisms of uptake of these particles into cells?
Yeah, we've done some of the studies where we've seen that some of the scavenger type of receptors have been involved. Especially the A type. I think some of my colleagues have a number of publications, revolving around these. But obviously, other receptors have also been shown to be involved, and the heparan sulfates, for example, and so on. So for the first interactions, yeah.
Thank you so much, Dr. Leto. We're all out of time for questions for this talk, but please come see him if you have any further questions. Thank you. That was a great talk. It is my pleasure to introduce Ainhoa Martinez from the Centro de Biología Molecular Severo Ocho.
So thank you, Crystal, for your presentation. As we talk about antisense oligonucleotide correct missplicing of neural microexon associated with autism spectrum disorder. So first of all, the author declared no conflict of interest. So we start with a short introduction about the disease. So autism is a disorder highly irritable that affects more than 1% of the children. It's characterized by deficits in social interaction and communication, linguistic development, and repetitive behavior. And in the minority of the cases are caused by single gene mutation or chromosomal abnormalities, and the most of the cases are idiopathic. So in this work, we could see that the brain of the patient with idiopathic autism show an imbalance in the CPB4 transcript exon 4 that result in a decrease of the microexon inclusion. You can see in the cardiology like an increase of the intensity of the second band that corresponds to the isoform in which microexon 4 is skipped. So the author of this work asks us that if it's possible to reverse this imbalance using antisense oligonucleotide. So the problem here is that the splicing mechanism that regulates the microexon is unclear, but we thought that maybe the intronic element proximal to the microexon can be responsible for their recognition. So the first strategy was to design a series of ECSO targeting to binding site of different silencer, upstream and also downstream of the microexon. And after the transfection in human neuroblastoma cell line, we don't see a significant effect, but we can see we identify an ECSO5 like a negative ECSO to promote the microexon exclusion. So because this is also a good result, because we generate at the first time a cell model of idiopathic autism. But because the aim of this work is to find ICSO to promote the inclusion of microexon and not the exclusion, we use another strategy. So in this case, we design a series of ECSO targeting to the 3' splice site of the downstream exon, so of the exon 5. And after the transfection, we observe that these 4 oligos promote the inclusion of microexon. So then we test different lengths to know the minimum site of the minimum length of the ECSO to favor microexon inclusion. And after the transfection, we observe also an increase of the microexon inclusion, but with lower efficiency compared to the 19 taken as a reference. So then we also check another and design another oligo, in this case, upstream and downstream of the 3' splice site to delimit the region in which the ECSO favor microexon inclusion. But in this case, we didn't see any effect. So now the next step in this work is try to know the mechanism by the ECSO5, the pathogenic ECSO, and also the therapeutic ECSO, what is the mechanism of this that promote the exclusion or the inclusion of the microexon. So in a recent work, this work, we said that we could see that the CPB4 is regulated at least in part by this protein, SRM4, that form a complex with 2 other proteins and bind to this motif to the intronic sequence upstream to the microexon. So when we analyze, when we analyze the sequence, the target region of the ECSO5, we see 2 similar motifs that could be responsible for the microexon 4 recognition. So to this purpose, we recently made an RNA affinity pull-down experiment in which we can know what type of protein binds to this region. So we first designed a series of biotin LA RNA oligos that by different steps that you can see in this figure, mixed with nuclear extract that contain all the splicing factor, and then the protein elution this protein, and then analyzed by Western blot. So we observed that the SRS11, that if you remember, form a complex with the SRM4, don't bind to this region, because we don't see any band, but we can see a binding of these 2 proteins, PTBP1 and HRMPC, that is describing the biography that regulates the microexon. So could be these 2 proteins could be responsible for the microexon 4 recognition. So then, in addition to this pathogenic mechanism, we also use minigene, in which we include the human CPB4 sequence from exon 2 to exon 6 and short introns, and with target mutagenesis. So we introduce a series of deletions covering the target region of the ECSO5, and also we make another mutant minigene in which all A from predicted branch point were changing to G or Ts. And after the transfection of this mutant minigene, we observe looking only the full length and the isoform in with microexon exclusion, that the microexon exclusion is increased in the mini-gene in which these motifs are partially delayed. So then, to continue with the mechanism with the therapeutic, the ECSO19 taken as an example, we also make a point mutation to increase and also to decrease the strength of the 3' splice site to confirm the kinetic model that is we decrease the binding of the splicing machinery to this region, maybe we promote the recognition and the inclusion of the microexon 4. So after day transfection, we observe that when the strength of the 3' splice site is decreased, the microexon inclusion is favored. So now continue with the effect of the therapeutic ECSO. We could see also in the first work and also in a more recent work of schizophrenia that when the microexon 4 inclusion is decreased, they are also associated with a decrease with poly-A tail length because the CPB4 regulate the translation of specific MRA by modulating their poly-A tail. So, and this is also correlated with a decrease of the level of the protein products of autism risk gene or schizophrenia risk gene. So here we try to know if the pathogenic ECSO have an effect of the level of the protein from a specific autism risk gene. So to this end, we use no human, if not a mouse neuroblastoma cell line to confirm the conservation of the mechanism between species, and with the idea to translate all this experiment to an in vivo mouse model. So first, we analyze the splicing profile after the transfection with the pathogenic and also with the therapeutic ECSO, but with a specific mouse sequence. And we observe the same effect, increase of the microexon exclusion for the pathogenic, and also an increase of the microexon inclusion for the therapeutic. So then we analyze by Western blot 2 proteins that are decreased in the patient with idiopathic autism and also in a mouse model with microexon 4. And in this case, after the treatment with the therapeutic ECSO, we observe an increase of the 2 proteins that's regulated by the CPB4. So now the question of this work is, okay, we have a strategy, but we can use this strategy like universal strategy to favor the inclusion of other microexon. So we test 2 microexon, TAF1 microexon and EA4G1 microexon that misregulate in neural disease. And using the same strategy, we observe after day transfection that both ECSO promote the partial inclusion of the microexon of the TAF1, but we don't see any effect of the EA4G1. So with all results together, we can conclude that on the one hand, we have identified an ECSO that induces CPB4 microexon exclusion, generating a cell model of autism, although more experiments are needed to understand the mechanism. And on the other hand, we have identified an ECSO as a potential therapeutic strategy for idiopathic autism and also for schizophrenia, characterized by CPB4 transcript isoform imbalance. Neural microexon inclusion may be promoted by ECSO targeting the downstream T' splice site, and our result broadened the application of the ECSO to treat disease characterized by altered inclusion of microexon. And so for the future, in vivo studies in autism mouse model will be provide the proof of concept of the therapeutic potential of the approach. So I would like to thank all results for the therapeutic ECSO are in this pattern, and I would like to thank to all the members of my groups and also the other 2 groups that participate and collaborate in this work, and thank you for your attention.
Great. Thank you so much. That was an incredible talk.
Thank you.
We can now welcome any questions to the microphone. Yeah, go ahead.
Hi, hello. Great talk. Two questions. First one, do you have any off-target with those ASOs? Because in these regulatory regions, will they be repetitive among most of microexons in other genes? And second question, do you keep isoform balance when you target these ones?
I don't take the off target, but I can, if I can remember, I think that maybe it's not any, because we also check if maybe this ECSO, that therapeutic ECSO, make an skipping of the exon 5, but we check this and we don't see anything.
I was referring off-targeting in other genes.
No check, we don't check yet. And the other, can you repeat, please?
If you have isoform balance, like if any other exons are affected by skipping this microexon 4?
I don't check in other gene, this imbalance.
In this same gene, in the same gene that you are skipping exon 4.
No, I don't see other, in this, in the CPB4 gene, only the imbalance that you can see here.
Okay, thank you.
Great, thank you. So sorry, we are out of time tonight for questions, but thank you. And any other questions can be directed after the session today.
Thank you.
Okay. With that, thrilled to introduce Joshua Robertson from UT Southwestern in Texas.
Hello, everyone. Thank you, Jillian, for that great introduction, and thank you, OTS, for having me. So today I will be discussing my work titled Synthesis, Characterization, and Biological Evaluation of Novel Three-Component Lipid Nanoparticles for mRNA Delivery. That was a mouthful. We're still working on the title. Oh, no disclosures, conflicts of interest, or anything. So just starting off, lipid nanoparticles are the most clinically advanced delivery system, as we see here from Alnylam with Onpattro, first FDA-approved LNP drug back in 2018. What I really would kind of point out here is that you see with this nanoparticle formulation, there are 4 components. There we go. You have your ionizable lipid here. You have your helper lipid, phospholipid, cholesterol, and PEG. And these are the 4 components that form the LNP. So a little bit more recent and also had great impact on our world today was the delivery of about over 5 billion doses of the mRNA vaccine made by both Moderna and also Pfizer and BioNTech. So as we see here, there are still those 4 components. However, if you look at the ionizable lipid, you see instead there's now the inclusion of branched allotails. And what my project is really focusing on is the inclusion of this hydroxyl group, as you see here and also here. So as I alluded to, my project is really looking at that hydroxyl group. As we see here from this molecular model, we see that amphipathic lipids and RNA molecules, they actually associate in water pockets. And what you're seeing in this model here, what you're seeing is this model here, these are all water pockets. And instead of mRNA, in this case, it's siRNA. And what was interesting looking at this model is you see a high inclusion of both the ionizable lipid and also the helper lipid, as you see in the magenta and yellow. That's kind of right zoomed in. And what was really interesting there, our lab was kind of looking at, okay, is it possible to combine aspects of both of those lipids into one to actually make a more simple LNP? So instead of 4 components, could we do 3 components? And sure enough, our lab was able to do this back in 2017, and we called this scaffold zooionic aminolipids, or ZALs. And they were designed to first increase molecular interaction with longer messenger RNAs, but also make a simpler LNP. So as you see here, back again. As we see here, we have the inclusion of the allotail, the helper lipid. We have the zooionic aspect, as you see here, the cationic aspect here, and what's really important for my project, that went out, is the hydroxyl group that's there. So with this scaffold, one of our main lipids that came from this scaffold was ZA3-EPI10, as you see on your screen here. This lipid is only 3 components that you see from the bottom, those molar ratios right there. And what was really interesting to see here is that it had really high delivery, and it was also multi-organ tropism in the case of the lung and liver, as you see there. Saying this is kind of really where my project started. So using ZA3-EPI10 as our scaffold, we really wanted to see how important is that hydroxyl group, and can it be changed to make a eventually a better lipid. So what we initially did is that we first synthesized ZA3-EPI10, as it hadn't really been touched since about 2017. So we started from scratch there, made ZA3-EPI10, and we then chose some substituents that kind of covered various parts of the chemical space. So we have the inclusion of the halogen with the chloride here. We have an opivylate that's more of a bulky ester, and also we have our acetate that is serving as our ester in this case. And we were able to synthesize these after a lot of optimization, and those conditions you see here are the most up-to-date conditions. So after we made these LNPs, we then wanted to incorporate them into 3-component lipid nanoparticles, and we did just that. We use vortex mixing for our in vivo studies, but for the sake of our in vitro studies, we actually use hand mixing, where we literally got a pipette and we just mixed it really fast for like 30 seconds. Thumbs kind of started hurting after the 3rd nanoparticle being made. So as we see here, the sizes are not really changing that much due to the changing of the substituent. And also I want to point out, so you see the red bar, that is 4A3-SC8. That is an ionizable lipid that was developed by our lab. It's one of our best lipids that we have, and it's actually a 4-component lipid. So we wanted to have a kind of a base to compare all of our 3-component results to. So again, the size was not really changing much. However, we did see stark differences in the case of RNA encapsulation, as we did a very normal ribogreen. This is a kit you can buy online. It's pretty easy to do in that regard. So what we see here is that 4A3-SC8 has really high encapsulation, as expected, because it's one of our best performers. ZA3-EPI10 also has really high encapsulation, which is really nice, recapitulating the work done by a previous student. However, we see a really drastic decrease with all of our derivatives. So we were looking at that, and we were kind of trying to figure out why would that be the case. But better yet, is like, is it still worth it to continue on with the project? We opted to continue on anyways with these LNPs, and we went to do in vitro studies. So we used IGROV1 cells, and this assay took about 2 days. We would plate our cells, we would do a toxicity assay, and then we would do luciferase delivery and use IGROV1 cells to do that. As we see from results here, of course, 4A3-SC8 performs really well. ZA3-EPI10 also performs very well. And there is still hope for the acetate group there. There's a big difference in the activity. However, it is present, and that was good enough for us. And also, we see from the chloride and the opivylate, there's little to no activity. So we didn't really have high hopes, but that's okay. The acetate gave us little hope. So from our in vitro studies, we went then to actually go and do our in vivo. As you see here, 4A3-SC8 performed really great. The ZA3-EPI10 was really interesting. So we actually see more specificity towards the spleen, which is really nice to see. The opivylate and chloride were pretty horrible, but that's okay. We kind of expected it from the in vitro results. But what was really interesting was looking at that acetate. So from the previous slide, I showed you that the encapsulation was quite low. However, as we see here, activity is really high, and it's also really specific to the spleen, which is really nice to see, because normally LNPs have been going to the liver previously, including 4A3-SC8 as well. As you see, it's really high in the liver. So this is really interesting to see here, because we may have actually found something better than the parent ZA3-EPI10 for delivering mRNA. And this was really, it's really a great find. We did all the work making all the compounds. We may actually have something that works really nice. So really some of the key findings here, what we found is that when you modify the hydroxyl group, number one, it is fairly important. And also when you modify it, there's the opportunity for more expiration. As we saw with some of the derivatives, they had really low delivery and almost no activity. But in the case of the acetate, it was really high delivery in vivo. And yes, this is also the second biggest takeaway, is that the acetate has hope, and it gives us more hope as we're developing other derivatives to be made, and that's what we're currently working on now. And with that, I really just want to acknowledge my lab. Also, Dr. Siegward, who is right there. That is my PI. He really just kind of let me take this project on as my own, make the kind of lipids that I wanted to make. I really go inside of our chemistry cabinet and see what compounds we had there and just try to do work. So it was really nice to be able to do that. And I would also like to thank my funding sources, as you see here. And of course, UC Southwestern as well. I really enjoyed being there. And with that, I have a little time for questions, if you have any questions.
Well, I'm surprised no one else is jumping up here to ask questions. Well, I'd love to ask a question. So normally in this field, first organ is the liver.
Mm.
Speculate on why you're getting it to go to the spleen and not the liver.
So as far as doing direct studies with my project, we haven't done that yet. However, previous work done in our lab actually showed that lipid nanoparticles form a sort of protein corona when they're injected. And with that, the different proteins that are actually on that surface have been seen to affect targeting to other organs. So perhaps in our case, when I eventually do my protein corona study, we may actually see the proteins that are more targeted towards spleen. But yeah, we haven't been able to do a protein corona, and we've seen there's different types of and different levels of proteins on the outside for targeting.
I have a quick question. When you bring the acetate and other functionalities, did you correlate that with the pKa of the amine head groups? Is there a major change?
That's a great question. So another previous work done in our lab actually showed that pKa can also affect the targeting toward different organs. And what we found from that study, that pKa's around between 9 and 11 are actually more spleen targeted. And the work that I actually did with my LNPs showed that they have pKa's in that 9 to 11 range, which would actually correlate toward their spleen delivery.
Very nice talk, thank you. I'm wondering if you plan to go back to the cell lines that you analyzed and analyze delivery in spleen type cell lines compared to liver type cell lines, for example, and to see if you get better luciferase delivery in these cell lines?
Yes, so that is definitely something we plan on doing. We started with iGrov1 because it's kind of one of the standards we use in our lab, and they're normally pretty hard to transvect. So if we can transvect those, hopefully we can do others. But no, that's definitely something we plan on doing, is trying to get organ-specific cell lines and see how they work there.
Are there any other questions, maybe online? Well, you did a very good job, Josh. Thank you so much.
Thank you. And I just wanted to say thank you again. If you have other questions, feel free to come by my poster. It'll be P203. Yeah, thank you.
Next up, it's my pleasure to introduce Cameron Thorpe from 11 Therapeutics.
Super. Thank you very much. It's a real honor to be here today at OTS to present to you the xRNA platform that we've been developing at 11 Therapeutics. My name's Cameron, and hopefully we can show you a bit of the next generation of RNA therapeutics that we want to develop. So before I start, and I just want to introduce that we actually think that RNA might be some of the worst substances for RNA therapeutics. And that's because when we look into recent drug developments and clinical trials, we find that there's not actually any RNA often in these drugs. If we look at this PMO-modified splice switching ASO, not a single 2' hydroxyl, it's no RNA nucleotides, and it's fully modified. Again, with mipomycin and RNA is H-activating oligonucleotide, we have 2' fluoro, DNA, phosphorothioates, but no RNA. With these interfering double-stranded RNAs, there's many, many modifications, including the addition of the 2' methyl. Again, no RNA. In fact, it's only with more modern mRNA vaccines, such as Spikevax and other mRNA developments, that we see unmodified structures. And this is where we want to really look into this space at 11. It's about modifying and bringing modified xRNAs to mRNA. And we really think that using specific modifications and adding modifications to these mRNAs can reduce immunotoxicity, improve stability, and overall the therapeutic effect. So before I show you the platform that we're developing, I actually want to show you the feasibility and the potential of these drugs. So in this experiment, what we did is we looked at an in vivo model and we synthesized some unmodified mRNAs. And we also took our xRNAs, this is our modified mRNA, and we just looked at basically the expression of protein between the two. And both at 6 and 24 hours, you can see that the modified mRNA has much more activity. And I haven't shown you it today, but it actually retains the activity much longer as well. So the platform that I want to show you is called Terra. It's the world's first DNA-encoded library platform, and we want to look at the structure-activity relationship between chemical modifications and our xRNA. This is the number of modifications, this is where we want to put the modifications, it's the type of modifications, it's sort of all in one. So I won't be able to talk to you about everything today, but the platform itself is broken down into 3 phases. The build phase, this is where we're building the DNA-encoded libraries, we're building the barcodes, we're building the xRNAs, and we're building the whole library in the platform ready for the next stage. The next stage is measure. This is looking at the mRNA itself. This is looking at its biological activity, profiling the mRNA, and then finally feeding this information into our learn phase, which is AI models, learning about all the data that we have, which we feed back into the next screen of the build phase to develop a better therapeutic. So the build phase is what I want to start with. This is how we generate our library. And the way we do it is just on normal CpG phosphoramidite beads using phosphoramidite monomers. And if you imagine that we split these beads into 2 halves, a left subpool in pink and green is the right subpool. And what we do is we take these 2 beads in separate and we add maybe a 2' fluoro RNA to one side and a 2' methyl to the other. Now I'm going to skip this step and I'm going to come back to it. But what we do is we then pull them together. So we have a mix. We have both the sub and the subpools together, right and left, and green and pink mixed together. And we separate them back out again. And what we do is we add another RNA nucleotide. So this time, when we pull them back together, we have a big mixture. So it's a linear step, but it generates exponential number of different molecules within the library. Now, the problem with this is if I give you one of these beads and I say, hey, what is this mRNA that we have here? You wouldn't know. It's modified and it's RNA, so you can't sequence it. So what we do is every time we add an RNA nucleotide here, we also add a DNA nucleotide. And we do this in situ, in presence of each other, and we do it using an orthogonal chemistry that we've developed. So it allows us basically to synthesize a DNA molecular barcode that we can sequence. At the same time, we can synthesize an RNA untouched. So the orthogonal chemistry itself, it took us about 2 years to develop, and it was quite a lot of challenges. So these are some HPLC traces of synthesis that we did. So when we started in October 22, there was a lot of side products and various other different problems that we had to overcome. But by April of this year, you can see that the synthesis of our orthogonal chemistry works similar to DMT chemistry, which is really great. So what we did is we synthesized a library of different barcodes and we sequenced them. So this is some sequencing data of our barcodes. And what I want to show you is basically how robust the platform is. So in blue here shows you sequences that are the correct length. So these are barcodes without any insertions or deletions. And in green, these are barcodes which do have some insertions or deletions, -1 to +1. So +1 would be an insertion. And what you can see is this is a logarithmic scale. So in our first go, we got about 90% of perfect barcodes, which is really nice. But by the time we get to April, you can see our orthogonal chemistry. It really relates to the quality of sequencing data that we have. And in this case here, we get more than 99% perfect barcodes, which shows that the platform's really robust and that the orthogonal chemistry works really well for synthesis. So just to sort of summarize what I've shown you there, I've shown you that the xRNAs have a real potential to be powerful modalities as therapeutics. I've shown you that our orthogonal chemistry works really well to control the synthesis of a barcode and an RNA in parallel without touching each other and damaging each other. And I showed you that our barcodes are really robust and they have really high fidelity. So the question is now is, can we use this platform that we started to build to develop RNAs and modify them and start profiling these modifications? So that covers the build phase. So now I want to look into measure. So now we're going to look at the mRNA itself. We're going to look at how well it performs, and we're going to measure it. So we took 2,000 xRNAs, so these are different sequences, At the moment, we're putting them in cells, but we can also do this in cell-free data. We take a biological assay and a readout for this, and we sequence it, and we generate various scores, but the two main ones I want to highlight is the translation score. This is basically how well the mRNA is translated into protein, and the stability score. This is how resistant to degradation the mRNA is. So I can't show you all the data, but what I can show you is some of the data that gives you a flavor about how robust our platform is. So we did a couple of repeats of the platform, and what this line shows is when an mRNA is thrown into the screen several times, it gives you the same result, really good reproducibility. And we can basically see here they're really good expressing proteins, and down at the bottom are ones that aren't so good. And that leads me on to my next graph. This shows us basically our hypothesis of controls before we started our screen. So before we started, we thought about, what makes a bad control? Maybe it has something that stops it producing protein, like a premature stop codon. And we chose some really good positive controls, maybe something from the literature that we knew would produce a high amount of protein. And what we did is this graph shows both in the early and the late phase, which are just different time points, is that we can match our hypothesis of controls to the actual data that we're getting, i.e. a good control is producing a lot of protein and a negative control doesn't. Finally, we really think there's some really good insights that we can get from the platform. So the stability score in this case is something that we've measured in our screen, and the minimum free energy is something you can calculate from literature values or open access software. And what we find is we have a correlation from this. So this allows us basically to infer maybe what is a good RNA before we even screen it, and we find that it might be possible to get other data like this, which allows us to basically not start from zero, but really choose really good RNAs right from the beginning. So in summary of the platform, I showed you that the xRNAs have really good potential to be good therapeutics, and that our platform has really good value to boost this. I showed you that our combined orthogonal chemistry works really well with a split pool design to generate an exponential number of molecules from a linear number of steps. And I showed you that we're able to produce barcodes and xRNAs in situ, and that our barcodes are really good for sequencing data. Obviously, this wasn't done alone. There's a big group behind us at 11 Therapeutics. So I just want to thank everyone back in Cambridge and around the world. And thank you guys for listening. If you have any questions, I'll be happy to take them.
So, thank you. So I was just wondering, you mentioned the size there, 100 to 200 nucleotides.
Yeah.
I mean, if you're making mRNAs, are you thinking about much longer molecules, or
Sure.
substituting parts of them, or?
Yeah, so I can't comment too much on the full length, but obviously chemical synthesis is limited to 200 bases, maybe less for RNA. This is something we're addressing in some of our other platforms. We also design our oligos so that they fit within the standard lengths of phosphoramidite chemistry. And our orthogonal chemistry actually works quite well, so it doesn't really impact the length of synthesis too much, which is really nice.
Over here. Hi, Cameron. Very good talk.
Thanks.
One question. Do you measure the stability, but what about accumulation of these modified RNAs and the toxicity that they might have?
Yeah, so we're doing some more in vivo data with some of our other platforms. This one hasn't, this is more the development of the screening techniques. We do look at the toxicity. It really does vary, but we get really nice results. From these xRNAs, we haven't run any of these for toxicology just yet. But it would vary on what modifications you use, and it really varies. I think what you would do is you would do your screen, you would pick the best candidates, and then you would do the toxicology of those later on.
Thank you.
That's okay.
We have time for a few more questions if there's anyone that would like to come up to the microphones at the front. I do have a question in the meantime. Yeah. Are you using modifications that are already established, or do you have any work kind of incorporating new and upcoming modifications into your platform?
Great question. So this is the really nice thing about the platform. If you can make a phosphoramidite, you can put it in the screen. So when we start the screen, yes, we'll pick very obvious and maybe 2'-fluoro, 2'-methyl, very readily available. But as we get more exotic, I think we'll probably start, and we will start making custom phosphoramidites to put into our screen.
I missed something very fundamental. When you bring multiple chemistries, each chemistry is optimized for a particular mechanism of action.
Yeah.
So how do you find for a particular, for RNAi, you may not be able to bring the phosphoramidate for chemistries, things like that.
Sure.
How do you dissect that at the end?
Yeah. Tell me if I answer your question. So in this case, we're looking at mRNAs. So it depends. We would have in mind the sort of modifications we want to use. Some might not be compatible depending on the chemistry that we're using for synthesis. Phosphoramidates, I think you would be able to use these based on the chemistry that we're using to synthesize them. So
I'm not asking about the mode of getting a material, the final function. Are you asking for RNase H or siRNA or splice switching? And how do you optimize within the frame of mechanism of action?
Yeah, so this is very much, it's split into two halves. There's the synthesis, which doesn't have much correlation to the application. We can synthesize a library for, I guess, any application. If you want to use it for siRNA, you could generate a library of siRNAs. If you want to use it for mRNAs, you can do that. The optimization then comes separately in the biological function. So you would have to generate first on a singular or a couple different RNAs, the assay, but then the translation into the platform for the whole library, it doesn't really get impacted by that. So you can basically develop your assay separately to the library synthesis and then bring them together.
On the left, yeah.
How are you accommodating potential biases that come from the synthetic scheme of different coupling efficiencies of, say, the modification or the bases in the final library product?
So we control this in several different ways. One of them is in the actual biological screen later down the line. The other one is often in the later down processes, we can use stuff like a UMI to make sure that we're not biasing, and we can look at the number of reads. So what we can do is we can calibrate our synthesis before. We get a rough estimate or a pretty good idea of any biases in the chemistry. And then we also have control libraries. So this is something that goes in before separately, and then we can compare the actual results to the control library as well.
And last question here, or two more questions on the middle here.
Maybe I missed that you've shown the stability and the translation score data, and I'm wondering that data is correlated. Have you ever checked that correlation of that data in various different cell types?
We are looking at the cell types now. We haven't looked, I guess we have a couple different cell types that we're interested in, but we haven't got fully into the different cell types to look at their correlation just yet.
And last question?
You said you're synthesizing mRNA, right?
Yeah.
How are you capping your mRNA?
Yeah, great question. Various different ways. Obviously, there's a lot of different commercial ways. I can't comment too much on how we cap it, but you can cap it or you can leave it uncapped. It really depends what you want.
Great. Thank you so much.
All right.
And thank you
Cheers.
for the questions. Next up, we have Arwaf Albari from Hudson Institute of Medical Research from Australia.
Thank you. Hi, everyone. Firstly, I would like to thank the OTS for having me here today and giving me the opportunity to present some of my PhD work on defining the immunomodulatory effects of gapmer antisense oligonucleotides, specifically on TLR7 and 8. It has been known for a while that synthetic oligonucleotide engage with nucleic acid receptors, and one of the main challenges facing RNA therapeutics is to avoid the unnecessary immune activation. As such, there are many cellular receptors which can recognize RNA therapeutics. For instance, we have here the cytosolic receptors RIG-I and MDA5, and CGAS as well, and also the endosomal toll-like receptors, specifically here TLR7 and TLR8, which both detect RNA degradation products and play a critical role in the defense against viral and bacterial infections. So again, it's not surprising that RNA therapeutics are interacting with these innate immune receptors, since exogenous nucleic acid recognition is just a crucial part of the immune responses to pathogens. Beyond activation, it was also demonstrated that TLR7 and TLR8 driven immune activation by therapeutic RNAs can be avoided by certain nucleoside modifications frequently used. For example, it was demonstrated that incorporating pseudouridine in mRNA or 2' or methyl modification in short RNAs reduced pro-inflammatory responses by antagonizing TLR7 and 8 activities. However, how these modifications operate to avoid the nucleic acid sensing is not fully understood. So my lab has previously found that fully 2' modified RNA oligonucleotides were able to inhibit TLR7 and 8 sensing in a sequence-dependent manner, with some sequences were even more potent inhibitors than others. Interestingly, we found that one sequence combining fully 2' or methyl modification and phosphorothioate backbone modification was even more potent inhibitor of TLR7 and 8. And also selected DNA oligos on phosphorothioate backbone were reported to have broad immune suppressive effects as seen with ODN A151. So at the start of the project, we were interested to see what happens if we mix base modifications such as 2' or methyl, 2' methoxethyl, and LNA modifications with DNA bases on a phosphorothioate backbone as seen in Gabmer ASOs. Our main concern was that these modifications may end up creating immune suppressive antisense oligonucleotides, which their inhibitory effect on the immune system would not be seen until having a concurrent infection. So my PhD work has been to define the immunomodulatory effects of 2' or methyl, methoxethyl, and LNA Gabmer ASOs on TLR7 and 8 sensing. So by screening more than 200 2' or methyl ASOs, we found that the majority of these ASOs strongly suppressed TLR7 while potentiated TLR8 sensing of the dual agonist R848 in a sequence-dependent manner. And after in-depth sequence analysis and targeted sequence mutations, we were able to characterize the sequence determinants of some of these activities on both receptors. Importantly, we identified a core motif. We identified here a core motif, the 2' or methyl CUU motif, limiting the immune suppressive effect of 2' or methyl Gabmers on TLR7. However, when we mutated that motif, it became much more inhibitory of TLR7 sensing. Similarly, for TLR8 here, we found that that motif was mediating potentiation, and when we mutated that motif, it just lost its activity on TLR8 sensing. So we speculated that these activities were not limited to the 2' or methyl Gabmer ASOs, and then we decided to expand our observation to include the 2' methoxethyl and LNA Gabmer ASOs. So similarly to the 2' or methyl Gabmers, the majority of 2' methoxethyl Gabmer ASOs inhibited TLR7, like with 95% of the ASOs tested, reduced TLR7 sensing for more than 50%. And then looking at enriched motifs in the top 10 inhibitory sequences from our screen, we identified this motif here, which we thought that it's possible for, or it could be at play in inhibition. So then mutation here of 2 bases of the motif in F5 was the mutation of 2 bases of that motif in F5 ASO was sufficient to impair TLR7 inhibition, which also was confirmed in a dose response experiment, and confirmed further the motif-dependent TLR7 inhibition seen with the 2' methoxethyl ASOs. Similarly, for the LNA Gabmer ASOs, we found that TLR7 inhibition was also predominant, with 85% of the ASOs tested leading to 50% decrease in TLR7 sensing. Interestingly, we observed that the best LNA ASOs preserving TLR7 sensing preferentially harbored a 5' terminal CC motif, suggesting that this motif could be at play in preventing TLR7 inhibition in the context of LNA Gabmer ASOs. So then to directly test the implication of this motif, we mutated the 5' end CC of ASO9. And as it can be seen here, that this mutation or this mutant variant significantly increased TLR7 inhibition, demonstrating further the sequence-specific modulation of TLR7 sensing by LNA ASOs. On the other hand, for TLR8 function, we found that MOE-Gapmer ASOs potentiated TLR8 sensing, but less frequently than what we observed previously with the 2'-O-methyl ASOs. LNA ASOs, on the other hand, were much weaker potentiators of TLR8, with no ASOs actually potentiating when used at the lower dose, the 100 nanomolar concentration. Interestingly, a significant proportion of LNA-Gapmer ASOs, in fact, were rather inhibitory of TLR7 sensing, as we found that 28% of the LNA ASOs inhibited TLR8 for more than 20%. So to further define the impact of the chemical modification of the ASOs WINS on TLR8 potentiation, we tested their effect on a series of ASO 663 variants, sharing the same central DNA region and differing only in the WINS modifications. Interestingly, while ASO 663 with 2'-O-methyl and 2'-methoxethyl significantly potentiated TLR8 in a dose-dependent manner, the LNA variant failed to do so. And we reasoned that this variability could be owed to different processing of the ASOs by indoor oxonucleases, predicting that TLR8 potentiation would be dependent on ASO degradation products by synergizing with its ligand, which could be hindered by the LNA modification. So that led us to test the modulatory activity of shorter oligonucleotides on both receptors, TLR7 and 8, which they could be degradation products from longer 2'-O-methyl Gapmers. And through systematic analysis of all the 64 possible combinations of 3-base oligos, we discovered selected trimers controlling TLR7 and 8 sensing, with GUX motifs strongly inhibiting TLR7 and the GIX motifs inhibiting TLR8, while the CUG and CGG potentiating TLR8 sensing. And please, for further details on these trimers, check our poster on Monday. So just to conclude, TLR7 sensing was broadly inhibited by Gapmer ASOs, independently of the chemistry used, with selective motifs underpinning these activities. Also giving the protective effect that we have shown with the 2'-O-methyl CUU motif and the 5' terminal CC-LNA motif, we anticipate that Gapmer ASOs can be rationally designed to limit unintended immune suppressive effects on TLR7, thereby retaining its protective function against pathogens such as SARS-CoV-2. On the other hand, TLR8 potentiation was strongly impacted by the chemistries of the Gapmer ASOs, presumably relating to their difference in resistance against nucleases. And now we know that selected 3-bases 2'-O-methyl on phosphorothioate backbone oligonucleotides were sufficient to modulate TLR7 and 8 function, suggesting that short degradation products from ASOs can directly interfere with these receptors. And also our team now has demonstrated the therapeutic potential of such immune suppressive 3-mer oligonucleotides and several in vivo models that will be presented in our poster on Monday. So at the end, I would like to thank everyone that was involved in the project, our collaborators and funding parties, and thank you all for attention.
So I noticed that your sequence which you use, you had 4 Gs in the gap for the Gapmer ASO, and typically those type of ASOs tend to form self-structures. So I was wondering, did you ever take a different sequence which didn't have those 4 Gs in the gap and see if you still have, I realize your recognition appears to be on a different side, but did you look for any self-structure or anything?
No, not, yeah. Not
It might be worthwhile to look at that.
Yeah, sure.
Hi, very nice talk and very interesting. So I just wanted to know, like, from what I understood is actually the structure of the RNA actually plays a role in, sort of eliciting this response. So have you ever seen whether the methoxyethyl or the LNA or the normal RNA in an in vitro level or even in a computational level? Have you seen if they look the same? Like,
yeah, actually, we just started like structural work to and also like to resolve the crystal structure of how these oligos bind to TLR7 and 8 sensing. But yeah, this is as far as I know.
oh, okay.
Beautiful talk, Arwaf. Thank you so much. Next up, it's my pleasure to introduce Tommaso Tabaglio from the Institute of Molecular and Cell Biology.
Okay, first of all, I want to thank the organizer for inviting me to share with you all the Natura platform. We have been working on it for the past 2 years, and I really hope this platform can help anybody here that wants to use it to find a new blockbuster in antisense oligo. So I guess this talk pertains to whoever answer yes to any of these questions. So we want to help people to find if their new oligochemistry or their next formulation of nanoparticles or their conjugation can really work better than the standard or can go in a new organ, in a new cell type. And that's what we had in mind when we created our platform. We wanted to create a platform that can test many antisense oligonucleotide species in a quick and cheap way with single cell specificity and in real time, of course, being as high throughput as possible. And most of all, easy to use for everyone. When I say antisense oligonucleotide, I mean oligonucleotides, and we focused on these 4 species mainly: siRNA, CRISPR with their guides, gapmers, and SSO. So, of course, I've seen poster, we have all seen the presentations. Some people check if their oligos are working by labeling their oligos and checking if the fluorescence is appearing in certain cell types, or the unlabeled methods are basically not using any fluorophore. They just check their target at the mRNA level or at the protein level. So they, of course, have advantages and disadvantages. We all know that. And we wanted to create a platform that is unlabeled, so we are free to see the native antisense oligonucleotide, but with the advantages of the label method, so quick, easy, and single-cell specificity. And that's how we came up with the Natura platform, which stands for Nucleic Acid Therapeutics Unlabeled Reporter Assay. And let's dig into it. It's nothing really new, but the Natura platform consists mainly of this gene that has 3 exons. So the main transcript that in all the cell lines we tested represent more than 90% of the transcript codes for GFP. It has a nuclear localization signal, so you see the nuclei being very green, and that's pretty much it. Your cells will be green. But if you modulate the inclusion of exon 2, which is called frame shifter, the cells will turn red with turbo RFP. And again, if you modulate the inclusion of exon 3, specifically 3A here, there's another acceptor splice site downstream that will produce luciferase. So that's it. Natura is not just a gene, but is a compound, library of tool compounds that can alter exon 2 inclusion and exon 3A inclusion. And going back, we designed these tool compounds to be gapmer splice switching oligos that we call AVA, siRNA, and a guide RNA for CRISPR-Cas9. So we didn't design just one per type because we know we want to be sequence independent. So we designed at least two oligos for each of the species. So 2 different SI with 2 different sequencing doing the same thing. Same for CRISPR-Cas9, guides, gapmers, and splice switching oligos. We also have a mouse, which we call Calimero, and is a mouse that contains the Natura gene in ROSA26 locus. So it is constitutively expressed and is a green mouse in which we can test whatever we find in vitro in vivo. We don't have time today to talk about CRISPR results and gapmer results, but we have those. You catch me at the coffee area and we can discuss about that. So I'll focus mainly on SI and SSO. And how is the gene behaving? Well, the gene is behaving as we expect when we transfect this gene, which, by the way, can be stably integrated because it's in a piggyback system. We see that the cells we tested are mainly green. But we have some spurious production of exon 2 skip transcript. So there will be a little bit RFP positive and a little bit luciferase positive. This is not a bug, it's a feature, as every person in computer science would say, and I'll show you why. If we want to screen if an siRNA chemistry or delivery system is working, you can just include the siRNA 1 against exon 2 and test in a Calimero system. And that's what we did. We have two different sequences of SI targeting this frame shifter exon, and we see that GFP is indeed going down. But what if, how do we know that is on target and we are not eliciting TLR receptors and therefore everything is going down? Well, if you remember, we have that residual RFP that is expressed, and we can exploit that. This is exactly the same figure you've seen before. It's from flow cytometry, and it's the GFP expression normalized for the untreated sample. And when we transfect the cells with the SI targeting exon 2, we see a reduction in GFP. But also if we dump in the media some G quadruplexes in DNA, we see that GFP is going down. That is clearly not an on-target effect. So we want to be able to flag this as a bad result. So by looking at the RFP instead and normalizing it against untreated, you can see that the scramble and the siRNA1 that is targeting only transcript containing exon 2 are not reducing the RFP, whereas the G quadruplexes are. So we would flag that as a bad hit. Okay, what happens then if we look at switching from green to red or from green to luciferase with splice switching oligos or guide RNAs? We'll talk about only the splice switching oligos for the sake of time, but this is what we see, always in flow cytometry, so no RNA extraction to be used. We see that GFP, upon transfection of this splice switching oligo called AVA1, is reduced over time, whereas turbo RFP is immediately going up. And here you can see that is not working, but you can see that we can really look into single cell and not just look at the average of what's happening in the average of the cell population. We can see that after 24 hours, we have a bimodal distribution of the uptake, and after 72, that gets a bit more normalized. So this is interesting. In confocal imaging, that's what you see. So you can also do confocal imaging, and you see that upon transfection of AVA1, you see upregulation of turbo RFP. And the cool thing is that you can do it also real time. So you can look at the kinetic, how fast your oligo is acting. And let's remember, this is functional uptake. So the oligo is not labeled. It's the actual protein produced thanks to the oligo that is measured. So is this working in vivo? Because that's what everyone wants to test. We took the Calimero mouse, we dissociated the liver, and we see that there are, of course, various populations that we are now labeling and characterizing, and you see that upon scramble injection, single injection subcutaneous of full 2-O-methyl phosphorothioate splice switching oligo, we see this amount of GFP and turbo RFP. But when we inject AVA1 fully modified, we see that some cell population starts switching to red. So we can further characterize who these cells are and work it out. This is not universal. So some cells in the liver, some cell populations will not switch at all. So it's quite precise. Does this work also with luciferase? If you remember, if we skip instead of exon 2, we skip with splice switching oligos or with CRISPR guides exon 3A, then there is another splice acceptor site downstream that expresses luciferase, and we call this AVA2. So it's targeting exon 3A, and we see that when we transfect the cells with AVA2, we see luciferase going up in vitro as well as in vivo. So this is a wild type mouse. The other one is injected with just scrambled SSO. So this is the baseline of your luciferase. And when we inject AVA2 with two different chemistries, sub-Q, we can observe in the liver area luciferase getting expressed. in differential ways, so we can compare chemistries, but these mice are still alive as we speak, so we are monitoring over time. No need to sacrifice. We can really monitor the time frame. So I hope I convinced you that this system has all the advantages of both label and unlabeled methods without the disadvantages. And I just wanted to share with you very briefly what we found with our first screening using this platform. So we decided to test if there is a combination of chemistries for SSO that works better than others. So we wanted to test 1 million combinations. That was not in our grant capabilities, so we tested hundreds. And we tested different combination of OME, LNA, and MOE. This was our library. We randomly put LNA, MOE, and OME, and we could test them in 4 cell lines, to mouse to human, at different concentrations, and we collected more than 5,000 data points. Each data point has at least 1,000 cells in flow cytometry, so no RNA extraction. It was pretty quick. And what we found out is that the best performing oligos, the ones switching from green to red the most, have most of all a pattern. So you need to place certain chemistries in certain positions more than the base or the, for example, melting temperature. As you can see here, we are about to publish, so I cannot disclose much, but the pattern 1 has more capability of skipping exon as compared to the monochemistry or with a second pattern of chemistry. And that translates into, this is a fold change in psi compared to the monochemistry, and we see that it can go up to 10-fold increase at certain concentrations. So same sequence, different chemistry combination can really do better. Also in terms of cell viability, so they are less toxic. Can we apply these rules to a splice switching oligo that is not within the Natura platform? So this is my last slide. We just took drisapersen, that is a splice switching oligo that is supposed to skip exon 51 in the DMD gene, and we incubated the cells with very low amount of drisapersen, and as you can see here in red, the switch is very minimal. But if we just take the same sequence and we combine OME chemistry with LNA in a specific pattern that we found thanks to the Natura, we can observe a 10-fold increase in the skipping of the exon. So we would say that the Natura gave something good to us. We're about to publish. I really hope this will happen here at the conference, that you have a cool chemistry or a chemistry combination or conjugation or formulation that you want to test, and you just come to me and you say, can I use the plasmid? And I will say, yeah, very happy. There you go. We are also recruiting, so if you are searching for a job and you like very hot weather, we wait for you in Singapore. And with this, I thank you for your attention, and I'll be happy to take questions.
Great, thank you. We're happy to take any questions from the audience.
Thanks for a great talk. I'm wondering, for the different sequences you measured, did you find that the patterns are conserved in terms of the ones that perform the best across the different sequences?
Yeah, definitely yes. The very interesting thing is that we designed some oligos in one chemistry that is supposed to skip exon 2 that work very faintly, so not very efficient, the sequence per se. But when we change the chemistry pattern, we get to the level of the best sequence. So I would say this was quite shocking for us, being that some sequences that we might test with our walks when we try to design an oligo and do not work with the right chemistry might start working.
Very cool approach. One question, Tommaso. Do you think your construct might bias the splicing mechanism? Like, will they induce like certain mechanisms that they are not natural?
You mean in the host organism?
Yeah, or in the plasmid, or yeah. Given that you are inserting some sequences in the intron, like before the exons.
Oh, so how we designed this plasmid is to take intron and exons and modifying them from mainly rabbit So the oligos that we design are not supposed to hit any mouse or human. So when you introduce the plasmid in your human cells, we tested that all the tool compound library that we use do not target the analog genes. I don't know if I answered the question.
Yeah. But I was wondering, like, if your plasmid itself would, like, in order to choose like the right sequences, will be a little bit biased just by the construct, like if they will affect like the normal splicing of that exon.
So it might be if you transfect a lot and you select the cells that are super green, of course, that will capture a bit of, modify a bit the endogenous splicing. That's what I would expect.
Okay.
But what we usually do is transfect the plasmid, sort for the GFP positive cells that are okay GFP, and then start performing your experiment with the tool compound. That we didn't observe any problem.
Thank you.
Okay.
Hi, that was great. I was curious, did you guys confirm that the different isoforms of those construct, the different spliced isoforms are like, equally stable, efficiently translated at the same amount, export, there's so many factors when the proteins you read out.
It's a very good question. So for example, GFP half-life is different from turbo RFP half-life. So your comparison must always be with, let's say, AVA1. That's the best practice. And then you start modifying it and you compare apple with apples.
Okay, yeah.
But it's very hard to find two fluorescent proteins that have exactly the same stability, exactly the same half-life, fluorescence. So it's a bit tricky, that part.
Okay,
But with the right controls, you can.
Yeah, thank you.
Thanks.
Reminder, we have time for, perfect.
Yeah, hi. There's ongoing debate in terms of importance of stereochemistry at phosphorus atom and phosphorothioates. Looks like your system might be very useful to answer those questions.
I really hope so.
Have you considered that?
Yeah, unfortunately, we don't have the capabilities of synthesizing stereochemistry pure antisense. But if someone is, I'm very happy to share the plasmid, to share the sequences so they start synthesizing. That's what it's for. I mean, it's for the, I really hope the community will adopt this because it's, I hate extracting RNA.
Since you have both mouse and the cell culture, this is actually the experiment to do.
Thanks for the talk. So I was wondering, because you have this new chemistry that seems to be more powerful, or at least a combination, and you are also injecting it in Calimero. Do you can elaborate or say something about the toxicity and the efficacy in vivo?
So yeah, it's a very good question because it was always our point to test side-by-side toxicity and efficacy. The way we can test toxicity is with just cell count through flow cytometer. So we're able to see that already the chemistry combination that we have has a bit more cells. So cells are more happy. It would be nice to see if it's because of less TLR activation or some other like specific hits, and we test that. In the mouse, we have the experiment now with AVA1. Monochemistry and combined chemistry, and what we did is try to replicate the Ionis paper on gapmers, so checking only ALT, AST at 48 hours, and the chemistry, the Calimero library is not toxic, so is not, for some reason, maybe the sequence is luckily not, or is okay, and the ALT, AST are normal, are within the wild type range. So yeah.
Okay, thank you. Thank you so much.
Thank you very much. Thank you.
Great, thank you. And for our final speaker of the Next Generation Scientist session, we have Alex Felix from University of Pennsylvania.
Okay, so first of all, I want to thank the organizers for giving me the opportunity to present our work here. I was really looking forward to attend this meeting. I'm very happy to be here. So today I'm going to talk about one of the therapeutic approaches that we are developing for STXBP1 related disorders. So what is STXBP1? The STXBP1 gene encodes for syntaxin binding protein 1, which is mainly found in neurons. And within neurons, it's located in presynaptic terminals, where it interacts with its binding partner, syntaxin 1, and also other components of the SNARE complex to facilitate synaptic vesicle recycling and neurotransmitter release. So basically, STXBP1 plays a crucial role in neurotransmission. An heterozygous mutation in this gene leads to STXBP1 encephalopathy, which is a rare neurodevelopmental disorder that affects 1 in 30,000 kids. And these kids basically suffer from severe to profound intellectual disability, motor impairments, epilepsy, and most of them are non-verbal. So, and what we know from the molecular perspective is that the disorder is caused by haploinsufficiency due to loss of function mutations that lead to the degradation of the mutant mRNA by nonsense-mediated decay, or by missense variants that produce a misfolded mutant protein that is degraded. So our therapeutic approach is based on the fact that 60% of human genes are likely to be targeted by microRNAs. And as you know, these microRNAs can bind to target mRNAs, normally in the 3' UTR sequence of the transcript, through the RISC complex. And this interaction between the RISC complex and the target mRNA can lead to the translational repression of that transcript by one of the mechanisms that I depicted in here. So our hypothesis is that STXBP1 is being repressed by certain microRNAs that are binding to the 3' UTR sequence. And what we want to do from a therapeutic perspective is to disrupt or prevent the binding of these microRNAs using steric blocking oligos in order to stabilize the transcript and boost or de-repress STXBP1 expression from the wild-type allele that all these patients still have. So we started our work by generating a STXBP1 patient iPSC-derived line. To do that, we took a blood sample from a patient with a splice site mutation that leads to the incorporation of a premature termination codon. And then we reprogrammed these peripheral blood mononuclear cells into iPSCs. And then through differentiation, we can generate mature and synaptically active patient-derived neurons. We also corrected this patient iPSC line using a CRISPR-Cas9 system to generate a corrected or isogenic control, which are neurons that have exactly the same genetic background as the patient, but with a corrected nucleotide instead of the mutation. One of the first things we did is that we confirmed the presence of haploinsufficiency in this patient line. So you can see that there's a 50% reduction in STXBP1 mRNA levels in the patient line compared to the corrected control, meaning that the mutant mRNA is being degraded by nonsense-mediated decay. And we also observe around a 40% reduction in STXBP1 protein levels in this line with no detection of a truncated protein, meaning that the mutant protein is not being produced. Then we perform microRNA sequencing in these mature STXBP1 patient-derived neurons. And in parallel, we perform a comprehensive in silico analysis across 10 different microRNA binding prediction softwares. So in this table, I'm showing the results after cross-referencing these two data sets, where here we were able to identify microRNA candidates that are both highly expressed in STXBP1 patient neurons and also highly predicted to bind to the 3' UTR sequence of STXBP1. Also, intriguingly, CLIP-seq data of AGO2, which is one of the main components of the RISC complex from human brain cortex tissue, has also shown like very prominent accumulation and binding of this AGO2 protein in this particular region of STXBP1 3' UTR, which corresponds to the same region where some of our microRNA candidates are predicted to bind. So this really suggests that this region is highly regulated by microRNAs in human brain. So to determine which of these microRNA candidates are actively repressing STXBP1 in a haploinsufficiency context, we perform antagomer studies in which we design a bunch of antagomeres against the different microRNA candidates. Basically, these antagomeres are going to sponge the microRNA. And here I'm just showing an example of these studies, focusing on miR218, which was one of our top candidates. You can see that the depletion of miR218 levels upon treatment with this antagomer 218 leads to a significant increase in STXBP1 mRNA in patient neurons. So this really indicates that, for example, miR218 is actively repressing STXBP1 in the patient context. Also, using an orthologous approach, we showed that the transfection of HeLa cells, which also express STXBP1 endogenously, with microRNA mimics corresponding to some of these microRNA candidates also lead to STXBP1 repression. So this was an additional proof that these microRNAs can actively repress STXBP1 endogenously. Since from a therapeutic perspective, we want to disrupt or prevent the binding of these microRNAs in the 3' UTR of the transcript, we and with our collaborators at Ionis perform high throughput screenings in wild-type NGN2 inducible neurons. And across the different microRNA binding sites corresponding to those microRNA candidates. So these studies led to the identification of several SBOs that were able to increase STXBP1 in a dose-dependent manner. And here I'm just showing an example of one of those high-throughput screenings. So some of these SBO lead compounds that we identified were targeting these microRNA clusters that I highlighted here in 3 different colors across the last coding sequence and the 3' UTR of STXBP1. So then to validate these candidates, we tested them in our patient-derived neurons. And as you can see, these SBOs targeting these different microRNA binding site clusters were able to upregulate STXBP1, and some of them in a very clear dose-dependent manner in a patient context. So these studies really demonstrate that these SBOs were not only capable of increasing STXBP1 abundance in wild-type context, but also in an haploinsufficiency context. Also, we have investigated the mechanism of action of these SBOs to determine whether they are acting through a microRNA disruption mechanism. To do that, we use the HeLa cell system that I presented a few slides ago, and we initially focused in this intragenic microRNA site. And we showed that, for example, when we transfect this miR3911 microRNA, which is targeting that site, with a scrambled oligo, we see repression of STXBP1 endogenously, and when we transfect with this intragenic SBO, we are able to fully rescue the repression mediated by 3911. However, when we repress STXBP1 with a microRNA that is binding to a complete different site, like for example miR218, which is binding to that site 2 in green, this intragenic SBO is no longer able to rescue the repression. So meaning that specifically this intragenic SBO is acting through miR3911 disruption in that particular site. We also perform very similar mechanistic studies, but in this case, in this site 2, so basically here, there are 2 potential microRNAs that are binding to that region, and we show that these can modestly repress STXBP1 as well in these HeLa cells endogenously, which corresponds to the red bars, and we show that this site 2 SBO was able to rescue the repression mediated by both microRNAs, either alone or even in combination. So this was another demonstration that this site 2 SBO is acting through microRNA disruption. And finally, this is an overview of our therapeutic pipeline. So we have completed this step over here in which we have identified lead SBO compounds that are able to upregulate STXBP1 in a patient context. But one of the challenges to initiate our in vivo studies was the fact that the 3' UTR sequence of STXBP1 is not well conserved between human and mouse. And that's why we generated an STXBP1 humanized mouse model in which we replaced the entire mouse locus of the gene with the entire human gene. So these animals are already available in our lab. And basically here I'm just showing some RNA and protein data from these animals from mouse cortex tissue, just to illustrate that there's no expression of the mouse transcript in the fully humanized animals, which have 2 copies of the human gene, but there's full expression of the human transcript. And also we show that the STXBP1 human protein was expressed in these animals, and there were no significant differences with the mouse protein compared to the wild-type animals. So this mouse model is already useful for us because it allows us to initiate our in vivo studies, and especially safety and efficacy studies. But our final aim is to generate a humanized haploinsufficiency model by knocking in a mutation into one of these 2 copies of the human gene, because that is going to be the ideal model of moving forward, not only to perform additional efficacy studies, but also to look for phenotypic rescue assessment. And last but not least, I want to thank our collaborators. I want to thank the entire team at the Center for Epilepsy and Neurodevelopmental Disorders, and especially my mentor, Ben Prosser, who is also the director of the center. And also I want to thank Nanthana, who is a master's student that has been working with me for the last few months on this project. And thank you very much for your attention.
Hello? I understand you're using ionis. So are these C8 chemistries with the phosphorothioates?
No.
What chemistry you use?
We are using 2'-methoxyethyl with full phosphorothioate backbone.
And for the last slide, what you showed, what is the particular microRNA you're using, and what kind of dose it requires?
In general, you mean?
For this animal model, what you are showing?
So in terms of concentration, we are for treating these iPSC-derived neurons, we do gymnotic uptake of the ASO. So we use around 3 to 10 micromolar or up to 20 micromolar concentration for this gymnotic uptake. In the case of in vivo studies, it depends if you perform the studies in neonatal mice or if they are adult mice, but we are in the range of hundreds of micrograms of compound, for example.
Is that 218? microRNA which you are going after? Which microRNA?
There are different microRNAs that we are after. So we have shown that we have identified at least 3-4 regions where we are able to upregulate STXBP1, and those regions are targeted by different microRNAs. So we are not only going after one particular one, but also others.
Thank you.
I have a question. Do you know if those microRNA has other target in the cell? And do you think by hiding one target, you will increase the degradation of the other targets?
That's a good question. So these microRNAs, so what we are targeting is the binding site, right? So we would be disrupting directly the binding of the microRNA to the 3' UTR of STXBP1. So in theory, that shouldn't affect other genes because we are not targeting the microRNA directly. So we wouldn't be dysregulating other genes.
The microRNA will be more available for their other targets.
Okay. Yeah, so that's something that we don't know. So we haven't seen that yet. But of course, we would need to do but it's a good point. We will need to just check, for example, in treated cells, if there's any upregulation of other targets for that microRNA. But that's a good point, yeah.
What is the advantage of targeting the microRNA binding site rather than the microRNA itself?
That's a good question. Thank you. So there's two main reasons. The first reason is because if we target the microRNA directly, for example, using an antagomer approach, we could potentially be dysregulating many genes, like many transcripts that particular microRNA is regulating or repressing in the brain. And the second reason is because by target, some of these binding sites that we have identified are being bound by different microRNAs, which means that if you design an SBO that binds to the binding site, you could be blocking potentially the binding of multiple microRNAs that are binding to the same site.
Hassan from UMass. Great talk. My question is more from a translational point of view. If I understood your data correctly, you're getting around 10 to 20% increase of the production of the... So how does that translate to patients? Like, what do you expect? Is that enough to reverse symptoms, or are you going to optimize chemistry to get better activity?
Yeah, that's a good point. So with some of the candidates, we have been able to see up to 70% upregulation in the patient context. So we are getting, so compared to wild type, would be more or less half the way, or a little bit more than half the way. And regarding how much we need to upregulate or go back to the wild type levels to be to get a phenotypic rescue, that's something that we still don't know for STXBP1. But there's some other groups in the STXBP1 research community that they have shown that that perhaps you don't need to reach wild type levels, and maybe just getting halfway through is enough for patients. But that's still something that we don't know for this disease.
Yeah, over here.
Yeah.
I'd like to really congratulate you and your team on this work. In 20 years of listening to talks on microRNAs. Yours is one of the very few that appreciates the complexity of microRNA action and has addressed it experimentally in a logical and thorough way. I do have one question. When I hear microRNA 3911, anything that microRNAs are in the thousands, I start to get a little bit wondering about how biologically relevant they are, and especially with this one, what the mechanism are. Could you elaborate a little bit more on 3911 and why you think it might be active and what you think it's doing?
Yeah, so there's not many things that we know about 3911. We know that it's only expressed, if I'm not wrong, in humans and primates. It's not expressed in mouse, for example. And we also know that in this particular case, so 3911 is being expressed from that same locus of STXBP1. So basically the 3911 locus is in the antisense or in the minus strand of the STXBP1 gene. That's why we call it an intergenic microRNA. So it's being expressed there. So it has the capability of binding to the same region in the sense strand, which would correspond to the STXBP1 transcript.
Okay, forgive me for asking one more question, but I think it's a generally important point for the audience. It would be interesting to know how many copies of 3911 you have. That also gets to the question that we had earlier about whether targeting the message itself. Messenger RNAs are usually present at much, much lower quantities than microRNAs. So you should have no problem directly targeting it. It won't release a significant fraction of microRNAs. But this idea of knowing your targets and knowing your active molecules is just something that...
Yeah, that's a good point.
Anyway, thank you. Fantastic talk and fantastic work.
Thank you.
Great talk.
Another one.
Just two questions. First one, maybe I missed it. Like in your humanized mouse model, is the microRNA expressed in the mouse? And because you change actually the 3' UTR and you say that it was not conserved, so is the microRNA conserved and expressed? Will it has the same seed and target your mRNA? And second question, like
let's go with the first one first. Yeah, thank you. So these microRNAs, not all of them are expressed in mouse. So, but some of our, actually we were kind of lucky because our top hits were targeting binding sites and microRNAs that are also expressed in the mouse brain. So in those cases, we are also seeing repression of the mouse equivalent microRNA in that same site. When we, for example, use, let's call it mouse equivalent ASOs, and we inject them in a wild type mouse, for example. Yeah.
Thank you. And second question. So some of the missense mutations in STXBP1 disorders, they cause problems with the folding of the protein and aggregation. So if you are planning to advance to clinical trials, are you going to test like for these mutations?
Yeah, thank you. That's a very good point. So most of the work that we have been doing is in this patient line that has a clear loss of function mutation. Now we are starting to test all these lead compounds in different patient lines that we have generated and validated already. And in some of those lines, we have of course a bunch of missense variants. So I will have an answer maybe in a few months to that question.
Thank you very much. Great work.
One last final applause for all the speakers this afternoon.
Great talk. Thank you, everyone, for your attention and great questions.